To get started on the digestive system, I wanted to highlight some of the key learning objectives that we'll see throughout this chapter. So, first of all, we need to be able to figure out what the key function is of each region of the GI tract. What's going on in the oral cavity? What's going on in the esophagus and the stomach and the small intestine and large intestine? So, what is it doing? And then we got to think about, okay, now that we understand the function, what are the anatomical adaptations that allow that part of the body to carry out that function? And then last, what are the things that I want to kind of weave in as we go through each of these different parts of the GI tract is looking at where digestion of each of the macromolecules begins. So if you think about something like this delicious cupcake here, right, you can consider that it's got carbohydrates, it's got lipids, it's got a little bit of protein that would be in the flour and maybe the egg for sure if it's not a vegan cupcake. So where is it that each of those things is broken down? Because they're not all going to occur in the same place. So I'm going to weave that in as we go. With this video, I'm really just going to get us through to the stomach. So I'll look at the oral cavity, we'll look at the esophagus, and then I'll end on the stomach. So perhaps the best place to start here is to look at the tunics or the layers that make up nearly every region of the GI tract. You can very much think about the GI tract as just one tube running through your body. And in fact, until the food or the partially broken down food chyme actually crosses from the lumen into your bloodstream or into the lymph, it's actually considered not part of your body. So until we've absorbed it, until it crosses into our blood or lymph, it's not part of our body. So it's considered external environment. And so with that, if you think back to 201, with the epithelial tissues, you may recall that they're the ones that are exposed to the outside environment. You may also recall that they line the GI tract. And so these are the four tunics that you need to know. The first that I want to discuss is mucosa. And so that's what you're seeing here. And if you look, this is actually simple columnar epithelial tissue, if that rings a bell. Oftentimes that simple columnar tissue has microvilli on it that assist with absorption, so increased surface area to allow for increased absorption of nutrients. So we'll see how that epithelial layer may vary from one part of the GI tract to a next. Another thing that you'll notice as you look at this mucosa is you're going to have glands. You're also going to have, so here's one gland that's shown here. You're going to have mucosal secretions, so mucus will be secreted here. Certainly in the rectum and the large intestine, you're going to see quite a bit of that to assist with lubrication and moving things along. In addition, you're also going to have some hormonal secretions that would come from this mucosal layer. So the mucosal layer wraps what's called the lumen, and the lumen is nothing more than the tube or the space that the food particles, the food stuff is going to be passing. So lumen, mucosa. Outside of that is going to be your submucosa. And so this is all submucosa here. And you'll see that there's a tremendously rich blood supply as well as a tremendously rich lymphatic supply. So we will eventually see that in the small intestine, that's where the majority of absorption occurs. And fats have to be taken in or absorbed in the lymphatic system, whereas digested carbohydrates and proteins, amino acids, can actually go right into the bloodstream itself. So outside the submucosa, you have what we call the muscularis externa. So it's like an external layer of smooth muscle. And so that's shown in this graphic here. And with almost all the layers, we are going to have a circular layer of smooth muscle and what we call longitudinal layer of muscle. You will always find the circular layer to be deepest or closest to the lumen. So that's this one. And then distal to that is going to be the longitudinal layer. And those layers of smooth muscle are going to assist with peristalsis, or allowing the food to move through your body. They're going to contract in a pattern that's going to ensure that food keeps moving in the downward direction, no matter what position you're in, no matter if you're upside down or if you are laying down in bed. That action of peristalsis and this involuntary smooth muscle will ensure that food keeps moving on down the pipe. And then last but not least is what we call the serosa. So here it is labeled. And what the serosa is, if you recall from 201, is it's a double layered membrane. So the serosa is wrapping, you can see one layer wrapping on the top and then one layer wrapping around the bottom here. And it comes around vessels and blood supply. You see that this forms into the peritoneum that we see that lines our abdominal cavity as well. While I have this picture up, it's a good chance to also point out the plexuses. So you should appreciate that after we swallow food, it's all going to be involuntary from there. So there's got to be a really robust, localized system of nerves as well as hormones that are going to help us to efficiently and resourcefully, we can say, digest food. And so these plexuses, you can see there's a myenteric nerve plexus that's shown here that innervates this smooth muscle. And then there's another nerve plex that innervates the submucosal region and can target the glands. So to say a little bit about the oral cavity, you don't want to discount its importance. It plays a huge role in being able to mechanically break down food. So mechanical digestion is the process of taking a large food particle and simply breaking it down in size, from a large food particle down to a smaller food particle. Enzymatic digestion or chemical digestion is one that is actually going to chemically break bonds. So to address the mechanical piece first, you have your teeth involved, you have your tongue involved, you have your hard palate involved. All of those parts of your mouth are going to be used when moving food around and getting it to come into contact with those teeth, with those molars and premolars that are going to work to grind. The incisors are going to be involved more with slicing or cutting your food. Think about biting into an apple or biting into a steak. And then the guys in the back, right, our molars and our premolars are really going to do most of the work in grinding and breaking it down into smaller particles. So I also want to shine a spotlight on the role of saliva and our salivary glands in the digestive process. So saliva plays a number of different critical roles. So not only does it help to moisten our food, if you've ever had dry mouth or been nervous and tried to eat, it's not quite as efficient. And with that saliva, you also do have some digestive enzymes. So you're going to have lingual lipase, which is an enzyme. So lipase is an enzyme that's going to work to break down lipids. The temperature of your mouth can also soften some fats. If you think about a fat like butter or peanut butter, right? When it enters your mouth, the temperature also helps to begin to melt those fats. So salivary amylase is the other key enzyme that you should be aware of. So amylase is going to be responsible for breaking down carbohydrates. So initially beginning to break down carbohydrates. You may have done that experiment as a child where your teacher had you suck on a saltine cracker. And over time, you could detect that saltine cracker, which has little to any sugar in it, would become sweet because you're beginning to break down the starches in that cracker into the smaller components, into the disaccharides, which are in fact sweet. Now, the release of saliva is importantly going to be controlled by your parasympathetic nervous system. This is the first that I brought in a specific function of the nervous system, and predominantly it's going to be your vagus nerve. So if you recall from AMP1, with the parasympathetic nervous system, you want to associate that with the processes of rest and digest. And so there's so many clinical applications of this, or just real world applications. I think many of us have experienced cotton mouth or dry mouth when we're nervous. Maybe we're anxious before a test or a presentation or a race, and your mouth gets dry. You get really thirsty all of a sudden. That's because with that balance of the sympathetic and the parasympathetic nervous systems, when you're nervous or anxious, the sympathetic nervous system dominates, and the parasympathetic nervous system is suppressed, right along with the vagus nerve. And the vagus nerve really is responsible for stimulating the release of saliva. So that's why we experience that dry mouth in those sorts of circumstances. I love this cartoon here of Pavlov's dog. One of the things to consider too is that the process of salivation, it does also have a reflexive component. So we can think about food. We can see food, we can smell food, and we can reflexively begin to generate saliva. So it's not that food actually has to enter our mouth before saliva forms, but the mere thought of foods can stimulate the parasympathetic nervous system and get that vagus nerve and the saliva to increase. What you're seeing here are specific glands, so ones that you'll need to know for the lab component. So we've got the parotid salivary gland. You have the submandibular salivary gland, which sits just inferior to the masseter. And then the last of the three key ones is the sublingual salivary glands. Now, these are, in contrast to endocrine glands, these are exocrine glands. They have ducts, and through those ducts, saliva will travel into your mouth. Whereas an endocrine gland is releasing a hormone into the blood, and those endocrine glands are ductless. The process of swallowing, or deglutition, is the fancy word for that. When you swallow food, so you can see a bolus of food here, there's a number of different critical steps that are going to be involved. One of which is that the uvula, which is shown here, this is the epiglottis, but the uvula, that's what you can see when you open your mouth and look in the mirror and look way back in the back of your throat. That little piece hanging down is your uvula. And what this actually does when you swallow is it seals and it closes off the nasopharynx, so that you don't get food going up into the nasal passage. Now, if you laugh or you're speaking, sometimes that uvula doesn't seal all the way, so you may experience that awesome sensation of getting food or something that kind of goes up your nose or goes up the wrong way. And all that's happening there, again, is that the uvula is not sealing. So when we swallow, that food is going to have to travel past a number of different sphincters. We're going to see this word sphincter a number of times because what these sphincters do, they are regions of smooth muscle that constrict. And so they're going to control the passage of food and chyme as it moves through the GI tract. So this is your upper esophageal sphincter. When that opens, the bolus can travel through your esophagus. And when the lower esophageal, also known as the gastroesophageal sphincter, opens, then that food will then travel into the stomach. A little bit more on the esophagus. It's one of the only regions that is not lined by simple columnar epithelial tissue, but rather stratified squamous. And if you remember stratified squamous, it's designed to handle high mechanical forces, high heat. It's actually the same in your oral cavity as well. But if you think about the frictional forces when we swallow food, the temperature of food may be hot, maybe cold, maybe you're swallowing, a mouthful of chips that are scratching your esophagus. So for all those reasons, you are going to have lots of layers of epithelial tissue, the stratified squamous, that serves as backup and as protection in the esophagus. Now, in the esophagus, you don't have some of the other protective features that we're going to see in the epithelial lining of the stomach. And so I bring that up because when that lower esophageal sphincter that divides the esophagus from the stomach, if this doesn't seal all the way, let's say you've had a big meal and then you lay down, or let's say that you've had some spicy foods that are kind of irritating that sphincter, what happens is you get acid reflux, right? You get heartburn. I think we're all aware of that. But the reason that fluid that doesn't bother you at all in the stomach bothers you when it's regurgitated up into the esophagus is because the esophagus is not meant to handle the low pHs of the stomach. It's meant to handle high mechanical forces, sure, but not low pH. So that's one of the reasons that you have symptoms when those fluids enter the esophagus. Moving on to the stomach, key functions here are, again, similar to the oral cavity, going to be mechanical digestion, so the physical digestion of food. It is a really common misconception that absorption happens in the stomach. And the only thing that is absorbed in the stomach is going to be alcohol, some drugs, but you don't have any nutrients actually absorbed into the stomach. The stomach is primarily responsible for storing food, and breaking it down into smaller components. And so in order to do that, one of the adaptations that we see in the stomach that allows for food to be broken down into yet smaller components is you're going to see an extra layer of smooth muscle. So if you recall in that previous graphic that I showed, we had circular muscle and we had a longitudinal layer of smooth muscle. If you look at this graphic, we are adding an oblique layer. So the oblique layer is going to be deep to the circular layer. So it's going to be deepest of the three layers of the muscularis externa. And if you think about the shape of the stomach, right, it's this really three-dimensional kind of awkward structure in our body. And so by having this third layer of muscle that comes at a different angle, so oblique, think about an oblique angle, that's why it's called that, right? The muscle fibers run in a different direction. So that's just going to allow for the grinding and the churning of your food in the stomach to be that much more efficient. Now, when food enters the stomach, we term it a bolus. But as food is processed in the stomach, we call it chyme. So what leaves the stomach is called chyme. So you can see the formal definition here. So if we take a deeper look at the microanatomy of the stomach, so we're going to see those 4 tunics again, and I'm going to hone in on some of the changes that we're seeing here in the stomach to allow it to serve its role. So you have mucosa, submucosa, muscularis externa, and then the serosa. As I've already noted, the muscularis externa has an additional oblique layer of smooth muscle, but I haven't addressed the mucosa of the stomach and what really makes that unique. So let's zoom in on this layer here. So this is all mucosa. It is quite deep compared to some other layers. So here we are, zooming in on this mucosa. You have a gastric pit, and you have a gastric gland. So the gastric pit are pockets where you can kind of imagine chyme being trapped. And deep to that, you've got these glands. And so glands secrete, right? You should always associate a gland with secretion. And within this gland, you have chief cells, parietal cells, and enteroendocrine cells. So we are going to see that there is quite a bit of hormonal regulation of digestive processes. And so an enteroendocrine cell is a very generic term to describe a cell in the gut, so that's what entero means, that has an endocrine function. So it's going to actually release some hormones, and you see some of these in the stomach. This is the first time we're seeing one of those formally. Now, chief cells and parietal cells, you should be able to distinguish which one secretes what. So let's elaborate. So chief cells are shown here in purple, and then the parietal cell is shown here as kind of this trident-shaped cell. So they're much broader than chief cells, and they tend to be at more superficial layers of the glands, whereas the chief cells tend to be deeper down into the gastric glands. Now, what each of these do, so the chief cells are going to secrete pepsinogen. So pepsinogen is an inactive enzyme. And the way that it's activated is in the presence of hydrochloric acid, right? So hydrochloric acid is going to significantly drop the pH. As the pH in this environment lowers, the pepsinogen is actually going to be converted to its active form, pepsin. So pepsin is called that. Most enzymes end in ACE, and they tell you what they break down. Pepsin is a bit unique because it's telling you that it instead breaks down peptide bonds. So if you recall the bonds that string together amino acids in a protein, those are called peptide bonds. And so this is the first time that we have seen protein breakdown. We saw a little bit of carbohydrate breakdown in the mouth, the salivary amylase. We saw a little bit of lipid breakdown in the mouth with the lingual lipase, but we didn't see any protein breakdown in the mouth. And in the esophagus, nothing is excreted. It's really just a channel or a tunnel for passage of food. But here, this is the first time that we begin to break down the proteins. And pepsin is going to be the enzyme responsible for that. So to recap, the chief cells secrete pepsinogen. The parietal cells, these are the ones that are responsible for secreting the hydrochloric acid, which is going to lower that pH. Other functions of the low pH in the stomach, not only does it work to denature proteins, so just the low pH in and of itself, that's excluding the role of pepsin. So we're going to denature those proteins. It's also going to break down the tough walls of things like vegetables. So plant cell walls, fruits, those cell walls can be quite tough, but with a lower pH, we can more efficiently break those down. In addition, we're going to see that the low pH is going to help to kill bacteria. So there is a little bit of immune function here. So what I want to end with here is to wrap up what it is anatomically about the stomach that allows it to be so good at resisting breakdown from the ridiculously low pH that we're going to see there. And so these three things are going to play a really critical role. First of all, it is epithelial tissue that's lining the stomach. And so epithelial tissue, we know, has high mitotic rates. So it'll be replaced about every 3 to 6 days. In addition, you have tight junctions between epithelial cells. And those tight junctions serve as an impermeable or relatively impermeable barrier to ensure that low pH acids in the stomach and the lumen don't cross through those layers and potentially upset deeper regions of your GI tract. And then last but not least is you have a very thick layer of alkaline or bicarbonate rich. So when you hear the word bicarbonate, you should think alkaline mucus. You're seeing here in this histology image, we're probably 400 times magnification here, but you're seeing this simple columnar tissue. And you're also seeing mucus secreting cells through here that are going to ensure that there's an alkaline barrier to protect the lining of the stomach from the acidity that's within the lumen. So coming back to our key learning objectives, hopefully you can walk through now and kind of check yourself. So what are some of the key anatomical adaptations in the stomach that allows it to be good at mechanical digestion? What sorts of chemical digestion do we see here? Where do we see lipids begin to be broken down? Where do we see carbs begin to be broken down? And where do we see proteins begin to be broken down? Thanks, guys.